This time, Sprechen Sie Science returns to discuss the scientific method.
As you know, the scientific method is defined as a systematic approach that is used by scientists to solve any problem. It is composed of 5 basic steps, the first of which is making an observation. Make an observation about something which sparks your interest, and in hopes to answer a question about this observation, you go ahead and formulate a hypothesis.
Your observations lead you to your hypothesis, which are assertions about how these factors fit together. Are they directly correlated or not? That leads you to your null and your alternative hypotheses.
You may have heard a hypothesis be defined as an if and then statement, but this is incorrect. An if and then statement is defined in science as a prediction, not as a hypothesis itself. It is a prediction based on your hypotheses. Your null hypothesis is going to state that there is no relationship between the two factors being observed, and your alternative hypothesis is going to state that one of them affects directly the other one.
After formulating your hypothesis, you're going to perform your experiment, and that's going to let you test whether your hypothesis holds or not.
It is very important when you design your experiment that you figure out a way to standardize your experiment. A standard experiment allows other researchers in other labs in other parts of the world to reproduce your experiment and come up with the same results. So the way you standardize an experiment is stating these are the units of measurements that are going to be used. These are the conditions that we use for the experiment, and so on and so forth. So you make sure you control everything else except for your 2 variables, which we're going to be defining in a little bit.
So in your experiment, you're going to have variables. Your dependent variable is what you're trying to make a statement about. It's what you're focused on. Your independent variable will be used to manipulate the experimental design so that you can make some kind of statement about your dependent variable.
So going back to your null and our alternative hypotheses, the null hypothesis would say that whatever changes are made to the independent variable, the dependent variable does not respond to these changes. And the alternative hypothesis is really saying that if you change the independent variable, the dependent variable will respond to these changes. And that is what we're trying to look at in the experiment. That is what it basically boils down to.
There's a convention that when graphing your data, you put your independent variable on your x-axis and your dependent variable on your y-axis, so that you can more easily track the changes in one as relates to the other.
When setting up an experiment, it is very important to include what we call controls. A control group or individual is one that does not receive a treatment. For example, if you are comparing the effects of different colored lights on a growth of a plant, your control would be a plant that receives natural sunlight, plant in which the color of the light is not tampered with. This gives you a baseline to fall back into and tells you what would happen if your dependent variable does not respond to any changes in the independent variable.
From all of this, you're going to collect your experimental data. The question you ask and the way you set up your experiment will dictate the way that you analyze your data. And from your analysis, you'll be able to draw a conclusion.
And this leads us to the 4th step, which is coming to a conclusion. We have 2 possible conclusions in the scientific method. You can reject your null hypothesis, meaning that you found some relationship between your independent variable and your dependent variable, or you can fail to reject your null hypothesis, which means that you did not find any relationship between them. meaning when you change the independent variable, the dependent variable either does not change or it does not change in a pattern with the independent variable. So if you reject your null hypotheses, your data ends up supporting your alternative hypotheses. If you fail to reject your null hypotheses, your data does not support your alternative hypotheses. However you say it, it is very important that you never say that you accept your alternative hypotheses or that you have proven your alternative hypotheses, because this is not the way scientific experiments are set up. The way science is set up is that you can disprove a hypothesis or you can find supporting evidence for it, but you can never prove a hypothesis. That's why there are so very few scientific laws in biology. Now, once a hypothesis has been tested many times by many scientists in many different places, and they have come up with the same result, that hypothesis might either stand alone or be joined with more hypotheses on the same topic, and you will eventually come up with a scientific theory on the same subject.
A theory is going to be a statement or a framework that describes basically all that has been observed, all that has been supported about a fact of nature.
Just remember that a scientific theory has a very large amount of supporting evidence behind it, and it is generally accepted by the scientific community as true.
So now you've got a good understanding of the scientific method. If you can explain everything that has just been explained here using just the diagram on this slide.
Okay, so you start by making an observation, which leads you to asking a question about this observation. In hopes of answering this question, you're going to formulate a hypothesis. Remember that you have a null hypothesis and an alternative hypothesis. Once you have stated your hypothesis, you're going to design an experiment that will allow you to test that hypothesis, and you're going to conduct this experiment. From this experiment, you're going to collect data, and you're going to analyze this data. An analysis of this data is going to lead you to a conclusion, which can either be to reject or fail to reject your null hypothesis. And finally, once this has been done enough times and enough evidence has been gathered, you would develop a scientific theory.